What I want to talk about is work I've done in inferring nature in physics, biology, and using machine learning. And to keep things interesting, I'm planning to phrase the conversation as if it's a match between humans and nature, where humans score points when they infer something new about nature, and nature scores points when it's making it harder for us and trying to keep us at bay. So you might think that since everybody here, so I'm talking to people, so I might think that we would be team human, but even in ancient Greece there were the skeptics that maintained that nature cannot be known. So maybe even in the audience there's like different teams. Excellent. So we started with this fundamental question: what are the fundamental laws, the fundamental axioms of the universe? And the thing is that we don't know. We don't know, and this is depressing because we start being very ambitious and trying to kick off the project of inferring nature off the ground, but there is no ground. There is no bedrock upon which to build systematically and trying to infer the rest of nature. This is in stark contrast with mathematics, where we have axioms that we postulate and then we take for granted as bedrock, and we start inferring. Alex was very nicely explaining how there are some prerequisites for picking those axioms. But then we use them as given, whereas we don't have that in science. There's no bedrock. It's more like a swamp that you're trying to bootstrap yourself out of. So why do I say that we don't have the fundamental axioms of the universe? It's because experiments suggest that there are four forces that describe interaction of matter with other matter: electromagnetism, the strong nuclear force, the weak nuclear force, and gravity. And the first three have been unified in a common framework that we call the standard model of particle physics. But the fourth one hasn't been incorporated into a common framework. This is why we don't have the axioms, because we cannot, in a unified framework, describe everything. And the reason for that is ultimately how they deal with information. The first three forces explicitly preserve information, whereas the fourth force, at least on face value, seems to destroy it. And since you can't have both simultaneously, it's very hard to put them together. It's very hard to put them together, but it's not like string theorists have given up. And they have been trying to resolve this information paradox, first introduced by Hawking, who incidentally was Priya's PhD committee. And what they have figured out lately, as of 5 years ago, is that information seems to be lost in our universe because of the presence of baby universes that constantly split off of our universe and then merge back onto it. And if you take into account those effects of universes splitting off of ours and merging to this one, you can actually reproduce this effect that information seems to be lost because you only have access to one universe. And there was a seminal paper by Maxfield and Maroff in 2020, and also our paper from last year that reproduces this effect. So in our paper we used topological quantum gravity. So we had the regular gravity you have in our universe, and we unified it and resolved Hawking's paradox, not with these three forces that I'm showing there, but with another force which was described by topological quantum field theory. So we did. So in this model, which is a rigorous and analytically exactly computable model, you had a common framework with gravity and non-gravitational forces, and you were resolving the information paradox. But so there is a lot of progress, I'm trying to say, but still, we unified gravity with a completely random other force, not with these three. So still, we don't have the fundamental axioms. And you might say that, oh, that's bad, but now I'm doing computational biology, so we don't need gravity for that. And if I'm saying that we understand the first three forces nearly perfectly, then why not make a simulation? Oh, by the way, so this lack of laws of physics is how nature keeps us at bay, poses the first challenge. Nobody gives us the axioms of the universe. But like I was saying, we don't need gravity. So how about doing the following thing, like discretizing space and putting the standard model of particle physics on that discretized space, and then try to perfectly simulate a cell. That would be super cool, because then we would be able to find initial particle configuration in the cell, then simulate a drug coming in and seeing exactly what effect it would have. So we'd be able to ab initio calculate drugs and stuff. So can we perfectly simulate a cell? And of course, there's nothing particular about the cell. Can we simulate the universe or parts of it perfectly? And unfortunately, there's a theorem that says that you cannot do that. It's the Nielsen-Inomiya theorem. And more precisely, it says that you cannot simulate the standard model of particle physics on a discretized space. And since the standard model of particle physics is our universe, that means that you cannot simulate the universe. The proof of that relies on two ideas. The first idea is that the standard model has chiral particles, which means that when a particle rotates, spins like this, it can move in only one of two directions, either this way or the opposite way. And you call this right-handed and this left-handed. Yeah, so those are the only two possibilities that it allows. And in fact, it's interesting that it prefers the left-handed particles. But those are the only two possibilities. And the second idea is something that we learn in undergrad in physics, that when you have discretized space, momentum is periodic. That means that that values of the momentum periodically are completely equivalent. Using those two ideas, you can prove that the standard model of particle physics cannot exist in discretized space. Because if you have a particle, a right-handed particle like that, then this positive momentum, since we're in discretized space, would be completely equivalent to being negative. But that means that this particle will be both left-handed and right-handed, but the standard model treats those particles differently, which is an absurd, and by reduction not absurd, we prove that this cannot be happening. So there's another way to look at this. So this answers the question of whether we live in the matrix, as Professor David Tong from Trinity College likes to phrase it. If we cannot simulate the universe, then this indeed could not be a simulation. We cannot be living in the matrix, because then CERN wouldn't be giving the data results that it is giving us. Yeah, however, it's nice to say that it's comforting that what I'm experiencing is not a simulation, but from a scientific point of view, it's depressing. It's another point for nature, because we cannot have perfect simulations of the fundamental laws as we understand them. Oops. Oh, yeah, so we don't know the axioms of the universe. Even when we do know them, we cannot perfectly simulate them. What's left then? Well, clearly, what's left is to do experiments. But they come with their own issues. And in particular, all of analyzing data, because the moment you get your data, you have to analyze them and interpret them. And then you run into the generalizability problem, which, of course, is like a cylinder. If you look at it from the top, it manifests in different ways depending on your perspective. And yeah, even in philosophy, people have looked into that, right? Even in ancient Greece, Sextus Empiricus, David Hume, that we heard about. Also, the rule following I like to think of as this generalizability problem. And yeah, so it consists of the following thought experiment. If you have an apple and you drop it, you see it fall to the ground, and if you repeat that 100 times, it keeps happening. But are you allowed the 101st time to conclude that it surely will be like the previous ones, that it will fall to the ground? In other words, generalizability is about arguing from observed cases, based on observed cases, about unseen cases. And in machine learning, we're interested in those cases when the unseen cases, we know a priori that they're bound to differ a bit. And yeah, so that's why I call it out-of-distribution prediction. Or as of late, in the context of foundational models, we refer to it as zero-shot learning. Yeah, so, and in that context, so in general, if we allow the unseen cases to be arbitrarily different than the seen cases, then clearly we cannot possibly conclude something about them. We cannot conclude something about what you haven't observed. And this goes by the name, essentially, of the no free lunch theorem. What it says more specifically is that an algorithm can only be better at finding a data-generating distribution only at the expense of being worse at finding another one, at inferring another one. So our tentative algorithm A will, on average, have the same performance than any other algorithm when you average across all problems. Yeah, so this is a final, another point for nature, which is leading by now by 3 points. So how are we pushing back against this generalizability problem? What can we do about this issue? Well, what we can do about it is, what comes to the rescue is using constraints from prior knowledge. That's the way to get off. So what the no-freelance theorem introduces is this trade-off between bias and variance. The way to get rid of it is to introduce prior knowledge constraints. And the way to do that is to start gathering a lot of data and start noticing patterns. For instance, you can impose the pattern that your model has to have sparsity, and therefore data sets that could not be produced by sparse models would be rejected, and that will allow your algorithm A to overperform other algorithms in the relevant cases that haven't been excluded. But of course, in biology, we're trying to push further ahead from that, and that's what the Human Cell Atlas tries to do. Instead of just using sparsity, we're compiling very comprehensive data sets of both observational and interventional data across many organs, diseases, donors, both for single cell and also spatially. I'm not sure if this has a pointer, but like here you can see that now we know that like what genes are expressed in this point versus what other genes are expressed in this other point of this section of the tissue. I should also say that the Human Cell Atlas is co-led by Professor Sarah Teichmann and Aviv Regev. And it's this effort of trying to map all the different cell types that exist in the human body. Because although all the cells in my body have the same exactly genome, they make different choices. And the cell in my eye decides to be an eye cell, and the cell on my skin, a skin cell. So they decide to focus on different parts of the genome and undergo what we refer to as differentiation. And in order to understand better human health and disease, we're trying to map out all the different possibilities that cells have to opt for. Yeah, so that's how we push back. We start gathering now biological prior knowledge and build models based on that. And that's how we start pushing back against nature by compiling, though this is a very big consortium and a huge effort in single cell biology. And furthermore, in my work in progress in the Lotflash lab, we're building this dis2p model, which stands for disentangled covariates to predict counterfactuals. And it lies at the intersection between disentangled learning, fairness, and causality. And our idea is that during the training process, we augment the data by matching the app with one another to produce counterfactual pairs. And then during the training process, we constantly do the 3 steps of abduction, action, and prediction, and penalize the training process. So during the training process, we're trying to predict counterfactuals and make a model that predicts them better. Yeah, in order to have a better performance for out-of-distribution prediction. Another thing, what comes to the rescue about not knowing the fundamental axioms of the universe is another idea from theoretical physics, which is the renormalization group, and which essentially says that phenomena at different length scales are siloed from one another. They don't, yeah, so phenomena at the subatomic level are not relevant for phenomena at much larger length scales. Yeah, more precisely, what it says is that a theory that tries to explain a phenomenon at a specific length scale should be phrased with degrees of freedom that are in the same length scale, and in that way, that theory we will manage to have it be elegant and have it be need a finite number of measurements in order to be predicted. Yeah, so imagine in the context of a disease spreading, clearly the relevant degrees of freedom to capture would be the bacteria and were discovered by Robert Koch. And then we would be able to build a nice theory of how disease spreads. Clearly, we could also try to do it with subatomic degrees of freedom, but we would build a huge monster that could do it, but it wouldn't be any better in doing so than if we had used the right degrees of freedom. Because ultimately, our immense success in designing drugs and understanding biology tells us that the tiny details in which gravity is quantized in this way or in another slightly different way couldn't possibly matter. That's what this theorem, the normalization group, tells us. At the other extreme, you could think of trying to explain how disease spreads by not allowing yourself to use the notion of bacteria and build a theory solely at the people's level. And you don't have to think to imagine that, because in medieval Europe, that's exactly what people were doing. They were using this now debunked theory about how diseases spread, the miasma theory, invented by ancient Greek physicians. Miasma in Greek means a pollutant. They were thinking of it as a cloud that settles upon the town and causes people to suffer, and then that cloud arbitrarily decides to move to the next town over. And yeah, they had built this theory, which was, you do weird confounders, you could make it be predictive. They surrounded the Pope with fire, and that protected him, but it was clear because it was keeping the sick people out, and not any weird cloud groups. So you could also build a theory like that, but you would have to constantly keep modifying it based on weird confounders and weird patterns that arise, because you have to capture the real things. And that is another thing that rescues us. To do biology, I don't need to simulate the standard model of particle physics like you have suspected. And indeed, so in biology, most of the time we end up using, most of our problems are combinatorial. They have to do about molecules and concentrations of the molecules in solutions. But we don't need to simulate atomic dynamics, sometimes molecular, but most of the time it's combinatorial problems, and for those you can reliably use simulations. For instance, in this other work on the package empty drops, we had there is the following very important problem in computational biology. So right now in biology, we can figure out, like I was telling you, like different cells opt for different choices, combinations of genes to express. So it's important to be able to sequence and figure out what genes each cell expresses. And in order to do that, you have to capture the cell and count all the molecules in it. But sometimes you're trying to capture it and you miss. And you capture next to it a solution, water, with some RNA and DNA molecules in that random water because those molecules leaked from cells or nuclei into the water. So sometimes you capture the cell, and sometimes you don't, but you think you have because you still capture the RNA. And that risks confounding downstream analysis, because sometimes you have a noise, and you can mistake it to be a real cell, and then start imagining that you have a new cell type. So it's a huge problem in computational biology. And there are some methods to deal with that. For instance, there is this famous algorithm that people use in computational biology that's called Cell Ranger. And it says that, oh, come on, if you capture noise, it must be so much less than a real cell that has a lot of RNA and DNA in it. So I'm just going to say that all the observations that have a lot of RNA and DNA are real cells, and all the other ones are just noise. But of course, if the signal-to-noise ratio starts being bad, you cannot make that distinction. And so what if you have a cell that has as much RNA as the noise? Then clearly you're either going to have too many false positives or false negatives. You need a better method to do it. And our idea was to go in the observations that have very few RNA and DNA molecules. So if it has 10 RNA molecules, that must be noise. There's no cell with just 10 RNA molecules. So great, so those are surely noise. And then if you put all of these together, you can create a profile of the noise. And then you can go in the cases where indeed they are ambiguous. And so here it's how many DNA molecules you have, and here how many RNA molecules you have. So in these ambiguous ones that are in the middle and you don't know, which they are, since you have profiled the noise, you can do a statistical hypothesis test, whether the observation is statistically close to the distribution you created for the noise. And if they deviate, you think of them as cells. That was our idea, which doesn't make parsimony assumptions about the expected cells. And indeed, in the simulation, we show that we're doing a lot better than Cell Ranger because, so what we did in this simulation is we took a data set that's processed by Cell Ranger. Cell Ranger thinks that the red ones are cells and the black ones are not cells. And we took 1,000 observations of the ones it thinks are cells, and we downsampled the number of counts in each one to simulate a small cell, which should be realistic because it's like a reduction ad absurdum again, right? So I'm saying that, oh, okay, competitor, I agree with you. And you're right. Then let's downsample that. Then you can see here that Cell Ranger does miss the green population of small cells, whereas we find it because that small cell still is statistically different than the noise. Yeah. So, yeah, excellent. And in other projects in the Deichmann lab, we're focusing on the following very important problem, which is, so in multicellular organisms, in order to function, and since by definition they have many cells, those many different cells need to coordinate their activities. For instance, if my eyes see something, the rest of my body needs to potentially go on a fight or flight mode, and so they need to communicate this signal that my eyes got. So there is this need of cellular communication, and how they communicate, at least one of the ways they can communicate, is by the sending cell excreting a molecule that we call the ligand. That ligand moving on until it reaches the receiving cell that has another molecule on its surface that we call the receptor, and it activates the receptor and causes something inside the receiving cell. However, some receptors are orphan in the sense that we don't know any endogenous, any ligands that bind them. Yet they're at very conspicuous places, for instance, in the part of the heart that makes it pump, or in the brain, and conserved across species. So they're not useless. They're being used. We just don't know how. And that, for one thing, makes us not know enough fundamental biology. For another thing, creates potential side effects that we don't understand for drugs, because they potentially activate those receptors. Yeah, also, like in designing new drugs that would potentially want to activate those receptors, would be important to find which ligands activate them. And of course, this has been done, has been attempted with protein structure, looking at the molecules and trying to see how they fit together. But our idea is to use spatial genomic data sets, for instance, what I showed there. And since we now know at each spot in this section, in this image, what genes are expressed at each spot, we developed a hypothesis test, and we see that ligands or receptors tend to be co-expressed. So the ligand is expressed where there is a receptor to hear it. So nature, in a sense, is not wasteful in communicating. It doesn't express a ligand in a location where it would be useless because there wouldn't be any receptor to hear it. Kind of like a tree falling in a forest where there's nobody to hear it. That doesn't happen. Yeah, they are co-localized in nearby neighbors. So right now we're developing a generative model for trying to find gene programs shared between cells. Yeah. So and in doing so, they'll be able to capture those ligand receptor pairs. So gene programs define the identity of a cell. Like I was saying, there's an eye program that makes those eyes in my eye cells. But there's also those shared gene programs that capture those interactions between cells, and that's what we're doing right now. And yeah, and that's caused another point, and now we're at tie with nature. And yeah, so in summary, this went by faster than I thought, I don't know why. But Yeah, in summary, like empty-Rose Multium, we developed this package for powerful and accurate cell calling. This DTP for causal modeling of covariate effects on genomic information and cell communication for energy for deorphanizing receptors. And yeah, I would like to thank the labs I'm in, and with such a great team, of course, I'm sure we'll score another point and put humanity ahead. So yeah, thank you so much.
Thanks, Stathis. Very, very interesting. So I'm going to take the prerogative and ask the first question. So one is a comment and then a question. I mean, there are fundamental laws in nature. We just don't know all of them.
Right.
I mean, it's an incomplete set.
Yeah.
Right? So, okay. And then I just wanted to clarify, right? So the thinking on the information paradox has evolved quite a lot. So I think Hawking himself kind of changed his mind, as you know, towards the end, right? So he had a very nice analogy, which I repeat quite often, but I think it's instructive, is that suppose you have the Encyclopedia Britannica and you look up New Haven and, 100,000 people population, whatever, then you put that encyclopedia in a box. So it's printed, we read it, we read the information, then you put that in a closed box, you burn it down. So then, but all the ashes from the burning of this are contained within that box, so nothing is left in the system. So what's really, maybe the way to think about the information paradox is that we just no longer know how the information is stored in that ash, and we do not know how to extract information from that ash. So the fact that the information may be there, but just coded in a way that we do not have access yet, might be an interesting way to think about it, because as you know, in the context of black holes, where that paradox was brought up, now we believe that there are potential resolutions to the paradox, right?
The most promising is this island conjecture that prompted those BB universe ideas that were also in. I should probably let you finish the question.
No, but that was just a note. I mean, we can discuss that offline. But I was curious, in one of the vignettes that you presented of experimental work and simulation work, so what are the attributes that are used to define cell versus non-cell? So these are the simulations in single cell. Yeah, here, single cell biology.
So the attributes. So what do you mean by attributes? Like.
What differentiates a cell from a non-cell? Right, you mentioned that.
So it's this idea that a cell should not be damaged, so it should be very well preserved. And trying to see whether you captured a well preserved cell, or you captured just something.
So the non-cell is just a failure.
So we're trying to capture it, try to fish it. And we just captured some solution with RNA in it.
Okay. Thanks.
So it's like mechanistic. What makes it.
Yeah. Whether it's intact or not, right? The information is intact. Okay. Great. So that was just a point of clarification. Questions?
I have a question about your discussion of siloing at different stages. And this is actually drawing on an example from Eric Winsberg, who spoke in the project, but is here. And I'm curious whether you see this as consistent with the kind of support of use you were making. He was looking at a material science project that was working on semiconductor materials, and it gets segregated into different scales. So where you have fractures and the bonds, you use quantum mechanical models. At a slightly larger scale, you use classical molecular dynamics, and then different models at still larger scales. But they can't be siloed because there are, in fact, effects that cross scales that have to be taken into account. And what they then do is they develop handshaking models that are more localized and connecting two theoretically incommensurable models, but for local interactions. And I'm wondering if you're seeing that as advancing the point you're making about the way siloing helps us, or as a problem for what you wanted to say.
Yeah, I guess if the length scales are not immensely different. So here we're talking about like, yeah, string scale with respect to the bacterium is a gigantic difference of length scales. Indeed, it's not a clear cutoff. It's not like if it's 10 times big in length scale, it's going to be 0, and if it's like 9 times, it's going to be completely different. But yeah, the renormalization group does explain when you have, this approach why we get renormalizable theories at low scale. Of course, the effective field theories are also immensely successful at describing things, and they can be predictive.
I just wanted to add to Joe Rouse's point that even in phenomenology with other physical systems, like if you look at cosmic scales and stuff, the scales are actually connected, but the challenge is bridging those scales, say computationally or observationally, and putting together a picture that includes the crosstalk between scales, right? So far it's been kind of modeling limitations that have really prevented us to, so they seem siloed only because we don't have the description to appropriately connect cause and effect across scales. Because I mean, it's a.
So gravity is a bit different. After all, like the field theories we have for gravity are not normalizable, renormalizable, except for string theory. Anyways,
string theories, right?
Yeah. String theory is more than normalizable. It's finite. So I'm sorry, I forgot what you asked.
Coupling of scales.
Right. So yeah, but ultimately, for all those simulations and that bridging of length scales you're talking about, ultimately you don't need quantum gravity to do that, right? So at the very least, there is a decoupling between. So there are different length scales.
In a meta sense, yes.
So like even we think of like Earth and like the solar system as being on different length scales, but like our planet is like has only a length scale of thousands of kilometers, and the sun is on millions of kilometers, just 3 scales of magnitude. Whereas with the Planck scale, it's a lot more. That's what I was trying to say. That the normalization group erases those effects about relevant operators.
Any other questions?
A short comment in the defense of miasma theory, because but the thing is first it wasn't medieval. It didn't end in medieval times because Johns Hopkins was built on the assumptions of miasma theory. But the link between the theories is that, for me, miasma theory was pointing to smells that go from sick people.
I see.
But the smells were made by bacteria. So nobody asked, what is smell coming from? If they would ask that, they could end up looking at bacteria, which we discovered 200 years before they were linked to disease.
Sure.
It's a much more complex story, much more
a good point that, the questions that we ask always sort of shape the connections between cause and effect that we can make, right? So if we don't ask the right questions, we never quite come to the causes, right? Tim?
So I know most of this was about biology and cells. I got stuck at the first slide. So I'm trying to understand how you're thinking about. You said, I've got these 4 forces, and these preserve information, and gravity doesn't. Newtonian gravity does. GR does. Preserving information is just code for being bi-deterministic. So the standard theories you have of gravity certainly preserve information. So I don't understand why you're picking gravity out and saying it has a problem. In fact, standard quantum mechanics is really a collapse theory, and collapse kills information. So I would actually put the way you read it in the textbook, the first 3 don't preserve information, and the last one does. So I just don't even understand how you're thinking about any of this.
I think you're referring to black holes in particular, right?
I was referring to black holes. Actually, in the context of general relativity, there's no Hare theorem for black holes. That has been taken as like.
But that's not
anyway, like to get to the other points that were more interesting that you brought up. So actually, like the reason why we say that those 3 QFTs preserve information is also the point you were bringing out the other day that they're unitary. So they explicitly preserve information in that sense. They're quantum mechanical, so the moment you do an observation, yeah, you have jumpers of wave functions. You could interpret it in a different way. The standard way would be based on, in physics at least, it would be based on whether they're unitary or not. And about quantum gravity, not just gravity now, there's this information paradox by Hawking. And in his calculation, he was seeing that, indeed, there are no correlations. The radiation, which brings us back to your first comment, that he wasn't seeing any correlations. The radiation emitted from the black hole was thermal.
Right.
And he was having a lot of ideas of how to try to fix those things. The baby universe idea was actually also his, but somehow the society moved away from that. And then he himself started thinking about whether information is stored at infinity. Those papers he published with Strominger a little before he died. So to come back to...
Let me just make, again, I don't want to get to just make a comment, because people say this stuff all the time, and I don't think it's correct. The no-hair theorems don't show what you just claim they show. Because they show that asymptotically, if you have an actual star that collapses, there will be traces of that even at the boundary in the geometry. In the same way as your ashes after you've burned your manuscript, there are traces there, right? The no-hair theorem doesn't say that different initial states go to the same final state, even exterior. Right. But that they asymptote to it. So it's not really relevant to this question of information. I mean, I just think there's a lot of confusion in this.
So I think you're getting at that theory that Strominger and Hawking published about the celestial holography and whether information is stored at infinity. I think that's what I'm getting by your use of asymptotically.
I think. Jakub, you had a question?
Yeah, but it might be, I don't know if I want to ask this question. Well, maybe it's too early. But the question would be...
Quick. Yeah.
Okay, about the fact that you want to generalize and that you think that the universe has axioms. So why would you believe that in the first place, that the universe has axioms and is axiomizable? But maybe that's something for a coffee break. I'm not sure. Maybe people will start throwing stones at me now. I don't know.
Yeah, exactly. Maybe cocktails even. I agree with Brandon, right? As coffee is not going to do it for that one.
Ultimately, I was using the term axioms as a theory of everything that explains all the fundamental interactions of particles. That's how I was using it. I guess since we see theories that do generalize, there's a belief that there is something common. We're not in the no free lunch zone. There is a law that we need to find out. Not all laws are fair game. Otherwise, we would be in trouble.
Great. Thanks very much, Stathis. Thank you very much for a very interesting presentation.